In this very exciting lesson, you'll learn how you can train and evaluate your segmentation model for identifying tumors in MRI data. You will learn about the UNet architecture and the procedure that we can use to successfully train a segmentation model. As you'll quickly see, working with 3D medical data provides some significant challenges, and you'll learn about some creative ideas to tackle them.
We'll first start by talking about representing MRI data. We want to be able to represent the MRI data in a form that we can input into the segmentation model. As you've seen, our MRI images are not just a single 2D image like X-rays. An MRI sequence is a 3D volume, here seen in the axial view. Furthermore, an MRI example will be made up of multiple sequences and thus will consist of multiple 3D volumes. We'll look at how we can combine these multiple 3D volumes into one 3D volume. To do so, let's pick a slice through the brain. This is a slice through the brain viewed on 3 different MRI sequences. The key idea that we will use to combine the information from different sequences is to treat them as different channels. We can say one of the channels is red, one is green, and one is a blue channel in the same way that we have 3 channels of an RGB image. The analogy of the 3 channels representing the RGB channels is mostly useful for us to visualize how we're going to combine the channels. There's nothing special about the number 3. The idea of using different channels also extends to when we may have 4 or 5 sequences, which can be represented with 4 or 5 channels. Once each sequence is represented with a different channel, what we do now is combine the sequences together to produce one image, which is the combination of all of the sequences. To us, this is now an RGB image. To the machine, these are channels stacked in the depth dimension. One challenge with combining these sequences is that they may not be aligned with each other. For instance, if a patient moves between the acquiring of each of these sequences, their head might be tilted in one sequence compared with the others. If the images are not aligned with each other, when we combine them, the brain region at one location in the red channel does not correspond to the same location in the green or the blue channels. So how do we fix this alignment problem? A pre-processing approach that's often used to fix this is called image registration. The basic idea with image registration is to transform the images so that they're aligned or registered to each other. We won't go into image registration in further detail. In the assignment, you'll work with images that have already been registered. However, it is useful to be aware of image registration as a useful tool when trying to combine 3D volumes.